AKT1 is a potassium channel through which plants absorb this essential element from the soil. It is composed of 4 pore-forming subunits that interlock in such a way that they form a barrel-shaped molecule with a pore in the center. At the opening of the channel, facing the cell exterior, there is a selectivity filter shown in black. The filter is composed of 5 amino acids per subunit, therefore we are looking at 20 amino acids and a potassium ion represented by the red sphere. When dissolved, ions are coated with water molecules that form a hydration shell. If 2 ions differ in size, but have the same charge, the smaller one will have a larger hydration shell. The mechanism of action involves replacement of the oxygens in the hydration shell of potassium by the oxygens in the carbonyl group of the amino acids comprising the filter. The distances are just right for the potassium ion, but not correct for the smaller sodium ion with a larger hydration shell. As a result, potassium ions are 10,000 times more likely to pass through the filter than sodium ions. Let us now examine the rest of the structural features of the molecule in order to visualize what happens after the potassium ions pass the selectivity filter. For better visualization, we will remove 2 subunits. There is a voltage sensing domain composed of 4 arginines. AKT1 is a voltage-gated channel, which means that it opens and closes based on the membrane potential. The phospholipid binding sites shown in red seem to be very important in modulating activity of the molecule through a yet-to-be-discovered mechanism. The third structural feature we would like to point out in this video are the serines located in the loop regions of the intracellular parts of the molecule. The channel is open when these residues are phosphorylated. Let us bring back the surface representation of the molecule, and bring back the other 2 subunits. We are now looking at the intracellular part of the channel. The potassium ion is visible on the other side of the molecule, but only because the intracellular parts are transparent. This is what may happen upon phosphorylation of the serines of the closed channel. The molecule undergoes a major conformational change from the closed state with a 2-fold symmetry to the open state with a 4-fold symmetry. The channel is now open, and the potassium ions can flow inside the cell. In order to make this video age well, it is important to note that although there is some data that indirectly points out to this scenario, opening and closing the channel upon phosphorylation is currently only a hypothesis. More research is needed to confirm this mechanism, and gathering relevant data is rarely an easy task. The cryomicroscopy structure you are looking at was not determined beyond 3.5 angstroms. Mass spectroscopy identified 2 phosphorylation sites per subunit, but only 1 site was visible. The other serine was invisible because of its location at the flexible region of the molecule. AKT1 is capable of sensing voltage, selecting potassium among other ions, interacting with phospholipids in the way that we still don't understand, and changing conformation from open to closed state and vice versa. What a serendipitous outcome of the countless random mutations over a gazillion years of evolution.